carbon dioxide transport, chloride shift, Haldane effect, three topics that almost every MBBS student finds confusing before exam. But don't worry, in the next 5 minutes, we'll divide the entire chapter using handwritten notes, flowchart, and exam focus point. And stay till the end because the last slide is a one-page revision checklist that can help you revise the chapter in just 30 seconds before entering the exam hall. So let's start. Let's begin with the basic concept. Carbon dioxide is continuously produced by tissue metabolism. The partial pressure of carbon dioxide inside the cell is about 46 mm of mercury. In the interstitial fluid, it is 45, and in capillary blood, it is 40 mm of mercury. That is, the pressure difference between cell and the interstitial fluid is 1, whereas between interstitial fluid and capillary blood is about 5 mm of mercury. This pressure gradient drives carbon dioxide from tissue into the blood. Remember, although the pressure difference is small, diffusion is very rapid because carbon dioxide is 20 times more soluble than oxygen. Remember this line. It is frequently asked in Viva. Now, where does all the carbon dioxide travel? That's our next slide. This slide is all about numbers. Total amount of carbon dioxide transported is about 200 ml per minute. That is, venous blood carries 52 ml per 100 ml, whereas arterial blood carries 48 ml per 100 ml. So, net transport is about 4 ml of carbon dioxide per 100 ml of blood. The distribution of this carbon dioxide transport is 70% as bicarbonate ions, the major form, 23% as carbamino compounds, and 7% dissolved in plasma. Remember this easy memory trick. Remember this number 7, 23, and 70. Always remember that bicarbonate is the most important transport form. Now, pause for a second and try recalling all these per 3 percentages before moving ahead. This is the core mechanism. Carbon dioxide enters the red blood cells. With the help of carbonic anhydrase, carbon dioxide combines with water to form carbonic acid. This carbonic acid dissociates into H plus and HCO3 minus ions. Hydrogen ions are buffered by deoxygenated hemoglobin, while bicarbonate moves into the plasma, and it is transported as sodium bicarbonate. Remember, mention this carbonic anhydrase and hemoglobin buffering to score extra mark. But once the bicarbonate leaves the RBC, how is electrical neutrality is maintained? Here is the answer. The problem that we have seen is when bicarbonate diffuses out, inside of the RBCs become less negatively charged. What is the solution? The solution is chloride diffuses in from plasma to replace this bicarbonate. What is the purpose? The purpose is to maintain electrical neutrality. This exchange is called as Hamburger phenomenon or chloride shift. So remember one simple line, bicarbonate out, chloride in. This one-line flowchart is enough for Viva. So what happens to the RBCs after the chloride enters? Let's see. Consequences of the chloride shift. When chloride enters the venous RBCs, total intracellular ions increases. This leads to the osmotic pressure rises inside the RBCs and some water is osmotically absorbed. As a result, the venous RBCs swell. The packed cell volume is slightly higher in venous blood than the arterial blood, and the venous RBCs become more fragile than the arterial RBCs. These are the classic Viva questions that students often miss. Remember, arterial RBCs when become venous RBCs because of the water osmosis, there is increase in packed cell volume by 3%. Now let's revise the remaining 30% of the carbon dioxide transport. About 7% is dissolved form. That is, venous blood contains 2.7 ml and arterial blood contains 2.4 ml. So net transported carbon dioxide is about 0.3 ml per 100 ml. And about 23% is transported in the carbamino form. That is, the carbon dioxide loosely combined with amino group in deoxygenated hemoglobin to form carbamino hemoglobin. Remember, deoxygenated hemoglobin binds more carbon dioxide. And this bond is reversible and loose bond which is easily released in alveoli. This concept directly leads us to the Haldane effect. The Haldane effect. Haldane effect is defined as oxygenation of blood in lungs displaces carbon dioxide from hemoglobin. And deoxygenated blood carries more carbon dioxide than oxygenated blood. In tissues, when oxygen leaves the hemoglobin, deoxyhemoglobin forms and more carbon dioxide is taken up. Whereas in the lungs, when oxygen binds the hemoglobin, carbon dioxide is released. So one sentence to remember, double the carbon dioxide carried from the tissues and double the carbon dioxide excreted in the lungs. That is, deoxygenation helps carbon dioxide loading whereas oxygenation helps carbon dioxide unloading. Also notice the carbon dioxide dissociation curve is almost linear, unlike the sigmoid oxygen dissociation curve. Now let's follow carbon dioxide into the lungs. When the venous blood reaches the pulmonary capillaries, oxygen diffuses in to form oxyhemoglobin. This oxyhemoglobin is a strong acid. It releases H plus ions, and to buffer this, bicarbonate re-enters the RBCs, while to maintain electrical neutrality, chloride moves out to the plasma. This stage is called as reverse chloride shift. When bicarbonate combine with H plus ions, it forms carbonic acid, which dissociate back or convert back to carbon dioxide and water with the help of carbonic anhydrase. And this carbon dioxide diffuses into the alveoli and is exhaled. Let's finish with some high-yield exam fact. Quick Viva revision. Whole blood is the ideal transport medium. Normal blood pH is 7.4. Respiratory quotient equals to carbon dioxide produced divided by oxygen consumed, and normal respiratory quotient is 0.8. These one-line facts are commonly asked during practical examinations. And now, the most important slide. This is your final revision sheet. Remember, total carbon dioxide transported is about 200 ml per minute. Net transport 4 ml per 100 ml of blood. Forms 3 forms: bicarbonate 70%, carbamino 23%, and dissolved form 7%. Key enzymes and proteins: carbonic anhydrase, which speeds up bicarbonate formation; bind 3 protein, which mediate chloride shift. The key effect is Haldane Effect, where the oxygen drives out carbon dioxide. If you can explain these points without looking, you are fully prepared for the university exams. And that's your complete carbon dioxide transport revision in just 5 minutes. If you need the detailed explanation and examiner-friendly answer writing, watch the main video. And if you want to remember chloride shift, Haldane effect, and the transport pathways effortlessly, don't miss the memory video. If this rapid revision helped you, like the video, share it with your MBBS friends, and subscribe to PhysioWithNeo. Remember our study formula: learn, revise, and remember. I will see you in the next rapid revision session. Keep studying.